Welcome back. In the previous lecture, Principles of Inheritance and Variation Part 1, we have covered the chapter till linkage and recombination. Now we are going to complete the remaining topics of the chapter in this lecture, Part 2. Now here I have kept some problems related to linkage and recombination. Let's try to solve them and then we'll go to the next concept, polygenic inheritance. So let's go to model number 1. So they are asking us like individuals who are homozygous for AB genes. Individuals homozygous for AB genes means AB and AB. They are crossed with the wild type which is plus and plus, plus and plus. So that means see, these are the parents. Now they are telling when this will make a gamete it will be plus and plus, and when this makes a gamete it will be A and B. Now you will get the F1 which is plus plus and AB. So this is called as F1 hybrid. The F1 hybrids are test cross. Test cross means crossing them with the recessive one. If this is a wild one, the recessive one will be AB with AB. So when the F1 hybrids are test crossed, you got the progeny of this and this in this ratios, and they're asking us to calculate the distance between A and B genes. Now Morgan worked on linkage and recombination. Morgan coined the term linkage, he coined the term recombination. Morgan's student has given a formula to calculate the recombination frequency, which will help us to calculate the distance between the genes also, right? What is the formula? Distance between the genes is equal to, the formula is distance between the genes is equal to number of recombinants by total number of progeny into 100. Distance between two genes can be calculated by using a formula, number of recombinants by total number of progeny into 100. So what are recombinants? If plus comes with A, that's a recombinant. If plus comes with B, that's a recombinant. If A is there with B, there's a normal one. If plus is there with plus, there's a normal one. So number of recombinants means it is 115 plus 105. So the formula, number of recombinants will be 115 plus 105, it will be 220. And then total number of recombinants, if you have to calculate, you have to plus 900 plus 880 plus 115 plus 105. So then it will come like 2200. It will be 2200. So then the formula will be 220 by 2000 into 100. So then it will be by 2000. So 11 you will get. So distance between the two genes A and B is 11 map units. So in this manner you can calculate one problem related to linkage and recombination. Let's go to model number 2. So this question is the exact question which appeared in NEET 2022 examination, including the numericals. So let us fastly see what is that question. So here we calculated the distance. There they gave the recombination and they are asking you to arrange A, B, C, D genes in an order. So the recombination frequency, here what you calculate is recombination frequency. By using recombination frequency you are telling the distance. Here they gave us the recombination frequency. The recombination frequency between two genes AC, BD, CD, BC, AB, AD is so and so %. Now they are asking what is the sequence of the genes in a linear chromosome. Is the sequence of genes ACBD or ADBC, DBAC, ABCD? They are asking us to calculate. Let us work out. So I'll take this option ACBD one by one, we will check and go. If it is like this, ACBD, so they told us in the problem A and C is 5. If I take A is here and this is C, A and C are separated by 5 map units. And then they told C and B, where is C and B? C and B are separated by, B and C are separated by 15 map units. That means somewhere here is B, this C and B are separated by 15 map units they told. And after B we need to write D. So and then they're telling B and D are separated by 9 map units. So this we have seen, A and B are separated by 5 map units we got. and B and C separated by 15 map units also be executed. B and D separated by 9 map units also we executed. Now let us see A and B are separated by 20 or not. Here is A and here is B. This is 5 and this is 15. So A and B, they're telling A and B are separated by 20 map. 5 plus 15 is 20 map units. So this also got tallied. Then they're telling C and D are separated by 24 map units. Here is C and here is D. They're telling C and D are separated by 24. 15 plus 9 is 24 map units only. So this is also working out. Then finally A and D are separated by 29. Here is A and here is D. They're telling that A and D are separated by 29 map units, 27% recombination. So let us total 15 plus 5, 20, 20 plus 9, 29. So 29 is also working out. That means the answer is nothing but the first option. So the order of chromosomes, the order of genes on a linear chromosome is A, C, B, D. So this is NEET 2022 question. Okay, so these are the problems related to linkage and recombination. Take a screenshot, then we'll get to the next topic called polygenic inheritance. So the next topic is polygenic inheritance. After linkage, the next topic is polygenic inheritance. Mendel, the father of genetics, worked in pea plant. He worked on contrasting traits. So he worked on 7 characters. If you take the flower color, he worked with violet color and white color. If you take the height of a plant, he has taken tall and dwarf. Means whatever he has done is he worked on contrasting traits, opposite characters. If you see, Mendel worked on violet color and white color, but if you go into nature and see, it's not just only 2 colors you find a gradient, right? We find different shades of violet, we find different shades of other colors also. That means there are some traits which are controlled by more than one genes. Because of the presence of more than one genes, they appear in that gradient. They appear in different shades of dark, they appear in different shades of white, like that. So if a gene is controlled by, if a character is controlled by more than 1, more than 2, more than 3, we call it as a polygenic inheritance. Let us see the sentences what are there in NCERT. Mendel studies mainly described those traits which are distinct in alternate forms. For example, flower color you take, he has taken only violet and white. But if you look around, there is a gradient, right? There's a gradient of colors. For example, if you take human height, humans are not just very tall and very dwarf. Not only 2 heights, we have intermediate heights also. Such traits, like take human height, such traits are controlled by 3 or more than 3 genes. Since a trait called height is controlled by minimum 3 or more than 3, we call such traits as polygenic traits, and such an inheritance is called polygenic inheritance. Polygenic inheritance not only takes 3 or more than 3 genes into consideration, it also takes environment also into account. And one example for polygenic inheritance, just now we mentioned here, that is human height. The second one is skin color. Complexion is also an example. Another example is human skin color for that. So let us take human skin color for a better understanding of the topic polygenic inheritance. There are 3 genes called A, B, C. A, B, C genes will control our complexion, will control our skin color. Now A, B, C genes, since we are diploid, A, B, B, C, C is a possibility, right? So now the phenotype, our color is controlled by this A, B, B, C, C. If all 6 are capitals, then the person will be darkest. If all 6, small a, small b, small b, small c, if all 6 are recessive, then the person will be lightest. Capital A, small a, capital B, small b, capital C, small c, then the person will be of intermediate color. So that means here, how many dominant alleles are there? Depending on that, darkness or lightness is depending, right? The phenotypic appearance is controlled by combination of each allele. When you are adding one dominant allele, the color becomes dark. One more dominant, furthermore dark. One more dominant, one furthermore dark. The genotype, for example, all 6 capital, darkest will be the color. All 6 recessive, lightest will be the color. One dark, one small, one dark, one small, one dark, one small will be an intermediate color. So this is an example of polygenic inheritance. Who first observed polygenic inheritance? Obviously not Mendel, right? Mendel observed contrasting traits only. So polygenic inheritance was discovered by Coleridge in tobacco plant. And what are the other synonymic names for polygenic inheritance? Means it's also called Galton's inheritance because Galton worked on it later. It's also called quantitative inheritance because the quantity of alleles you are adding, the color is changing. So that's why it's called quantitative inheritance. It's additive effect. The number of dominant genes you are adding, the number of darkness is increasing. So it's also called additive effect, cumulative effect. All these are the synonyms of polygenic inheritance. And coming to the examples, here we gave one example that human height is one example. Then we went to the human skin color example. So human height, human color and human IQ, these are examples of polygenic. And coming to the chicken, poultry, egg size and egg number are the examples. Coming to cattle, milk yield and meat yield are examples of polygenic inheritance. Take a screenshot. I'll show you this. I'll explain polygenic inheritance with the help of a Punnett square. You can understand it better there. We'll take skin color to observe polygenic inheritance. Now, as we discussed, skin color is controlled by 3 genes A, B, C. So, capital A, capital B, capital C is one parent. And this parent will be black, darkest. And another parent, all small recessive. So, this person will be very light, right, or white. Now, when we see the gametes for this parent, it will be ABC gamete, and for this parent, it will be ABC gamete. When we cross these gametes, we get the F1 hybrids. The F1 hybrids will be A, B, B, C, C. Now, this is intermediate color. F1 hybrids are intermediate color. Now, F1 hybrids are selfed, means this with the same thing, A, B, B, C, C. If the F1 hybrids are selfed, we will get F2. So, how many types of gametes we get when all 3 are heterozygous? I'll tell you a formula. It is 2 power n, and n is number of heterozygotes. So here this all 3 heterozygotes mean 2 power 3 means 2 into 2 into 2. 2 2s are 4, 4 2s are 8. Means 8 types of gametes we get here. 8 types of gametes we get here. Let us see what are the 8 types of gametes. Did you understand till here? So I'll show you. I'll erase this and we'll get into the 8 types of gametes to see the F2 ratio. So what are the 8 means? If you take A, B, C, then the 8 types will be all 3 capital, right? Then here we will write all 3 capital. Then 2 capital we have to go, AB capital or BC capital or AC capital. The other one will be small, C is small, A is small, B is small. Then the other 4 types of gametes will be 1 capital only, A capital, B capital, C capital. The other 2 will be small, B and C are small, A and C are small, A and B are small. The last gamete will be all 3 small. So we told 2 power 3 means 8 types of gametes will be formed. 1, 2, 3, 4, 5, 6, 7, 8. 8 type of male gametes, 8 type of female gametes, eggs. We will write 8 type here and 8 we already wrote here. So these 8 types will be A, B, C. Again, capital A, capital B, small c. Small a, capital B, capital C. Capital A, small b, capital C. Then small a, capital B, small c. Small a, small b, capital C. 1, 2, 3, 4, 5, 6. Small c, capital A, yeah, we need to write capital A, small b, small c. Capital A, small b, small c. Small a, small b, capital C, and small a, b, c. So 1, 2, 3, 4, 5, 6, 7, 8. Now we need to draw the Punnett square. Let us fast draw the Punnett square. So we'll get a 64 combination here. Instead of writing the genotype, which is time consuming, we can take the numbers also and we can proceed and we will get the ratio, right? So how we will take is, we will take 3 capital, all 6 capital. Instead of writing it as A, B, B, C, C, I'll write it as 6 dominant. I'll write it as 6 dominant. Understood? So 1, 2, 3, all 6 are dominant. Here, this with this, 1, 2, 3, 4, 5 dominant. For 5, I'll use black color. So 1, 2, 3, 4, 5 dominant. So 1, 2, 3, 4, 4 dominant, right? For 4, we will use another color. For 4, I'll use green color. 1, 2, 3, 4, 5 is there. So if 5 is there, I need to use 1, 2, 3, 4, 5. 5 we are going with black. So 1, 2, 3, 4. 4 we need to go with green. So 1, 2, 3, 4 with green. 1, 2, 3, 4 with green. 1, 2, 3. For 3 we will use red color. Like this we are counting, right? Again, here we will see 1, 2, 3, 4, 5. 5 we are going with black. 1, 2, 3, 4. 4 we are going with green. 4 we are going with green. So 1, 2, 3, 4 with green. 1, 2, 3, 4 with green. So 1, 2, 3. For 3 we are using red. For 3 we are using red. So 1, 2, 3 here, 1, 2, 3 here, and here it will be 2 only. So for 2 we'll use another color. No, I don't have other color. So for 2 also we will use blue only, right? So then again, so come to the third one. So 1, 2 capital, 3, 4, 5. 5 we are using black. So 1, 2, 3, 4. 4 we are using green. So here 1, 2, 3, 4. Again 1, 2, 3, 4. Here 1, 2, 3. So 3 goes with red. I'm just counting the number of dominants. Again 3, again 3. So 1, 2 only will come. For 2 we are going with blue. Then come to this one. So 1, 2 capital, 3, 4, 5. For 5 we are using black. And then next again, how many capital? 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. Then next here it will become 3 because 1, 2, 3 dominant. Here also 1 dominant, 2 and 3 dominant. Again 3 dominant, and again here only 2 dominant. All these are recessive. So 2 dominant will go with 2. Coming to the next one, here we have only 1 dominant, 2, 3, 4. So for 4, which color are we using? Green. And here 2 plus 1, 3. So for 3, which color? Red color we used. So this is 3. This will also be 3. This will also be 3. And here 1 and 2. For 2 we are going with blue color. So 2, 2, 2. So 1 only will be dominant here. For 1 we will use again black. Now next one, again B is capital, 1, 2, 3 and 4. 4 will go with green. Here also 1, 2, 3, 4 with green, right? And then all 3's will come because these 3 will be the same combination. 3, 3, 3, 3, 3, 3. Then next it will go with 2 only. 2, 2, 2, 2, 2 dominant, 2 dominant. And then last one will go with 1 only, 1 and 1, because here only 1 is dominant and here nothing is dominant. 1 and 1. Last line if you see, all are small there. So 3 dominant. For 3 we are using red. And here only 2 dominant. For 2 we are using blue. Again blue, again blue. Then here it is 1. For 1 we are using black. 1, 1, 1. And here all no dominant, 0 dominant. So like this we have to fill the combinations. Instead of writing A, B, B, C, C, it's time consuming. I just wrote the numbers here. Now we need to count how many 6 are there, how many 5 are there, how many 4s, how many 3s, how many 2s, how many 1s and how many 0s. Come on, if we see, take a screenshot of this. I'll erase a part of this. Right. Now if we see, how many 6 are we getting? Only 1 6 we got, right? This is the 6. So all 6 dominant You got all 6 dominant, you got 1. How many 5 dominant you got? So 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Somewhere we went wrong. 6. 1, 2, 3, 4, 5. This is also 5. Yeah. 6 dom, 5, 1, 2, 3, 4, 5, 6 we got. So 6 dominant, 1 we got, 5 dominant, 6 we got, then 4 dominant. How many 4s we got? So the 4s are here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So we got 15 4s, then 3 dominant. For 3 we use red color, right? So how many 3s are there? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So 3 dominant, 20 you got. So 2 dominant means blue color. So how many 2 we got? 1, 2, 3. 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 we got. So 2 dominant, again 15 we got. 1 dominant, how many 1s are there? 1, 2, 3, 4, 5, 6. So 1 dominant we got 6. And how many 0 dominant we got? 0, only 1 got. So that means the ratio what we got is 1 is 2, 6 is 2, 15 is 2, 20 is 2, 15 is 2, 6 is 2, 0, right? So here if you see 1, 6 dominant will be very dark, it will be black, and this one will be very light, and 20 will be intermediate. So this one, this is black, right? So this one will be very dark, and 4 dominant means fairly dark, and 2 dominant means fairly light, and say here 1 dominant means it is very light and light. So these are the shades we get. This is an example of polygenic inheritance. Take a screenshot. We'll get to the next topic, which is called as Pleiotropy. The next topic is Pleiotropy. So what is Pleiotropy? We need to understand. We have seen so far that one gene will control one phenotype. Means, for example, capital gene will control height of a plant, like that. Generally, one gene will control one phenotype, but there are instances where one gene shows many phenotypes, but there are instances where one gene exhibits many phenotypes. Then such a concept is called as pleiotropy. One gene usually shows one phenotypic expression only, but if one gene can show many phenotypic expressions, then that's an example of pleiotropy. A very good example for pleiotropy is phenylketonuria. There's a disorder which is called as phenylketonuria. Phenylketonuria, it's an inborn error of metabolism. It's a genetic disorder. It's an inborn error of metabolism. This disorder is due to mutation in a single gene. It is a single gene mutation. Phenylketonuria is a single gene mutation. Mutation in a single gene. What happens if that gene becomes defective? So then it's not able to produce an enzyme called phenylalanine hydroxylase. Because of the gene getting mutated, so that gene is unable to synthesize an enzyme. What is that enzyme? The name of the enzyme is phenylalanine hydroxylase. Now this enzyme, what is its role? Phenylalanine hydroxylase is an enzyme which is responsible for conversion of phenylalanine to tyrosine. So what happens if this enzyme is made? Then the phenylalanine molecule, it's an amino acid. phenylalanine molecule gets converted to tyrosine, and tyrosine will be further converted to melanin. In this manner it happens, right? Now if there is a defect in the gene which is coding for phenylalanine hydroxylase, then phenylalanine hydroxylase is not synthesized. If phenylalanine hydroxylase is not synthesized, then phenylalanine cannot be converted to tyrosine. If phenylalanine is not converted to tyrosine, then tyrosine is not made. So what are the effects we will see? No tyrosine. So if there is no tyrosine, then phenylalanine gets accumulated, right? The phenylalanine gets accumulated. Where does the phenylalanine gets accumulated? It gets accumulated in the brain. So since there is no tyrosine, phenylalanine accumulates as phenyl acetic acid and phenyl pyruvic acid. Phenylalanine gets accumulated as phenyl acetic acid and phenyl pyruvic acid. Where does it accumulate? In the brain. Since it gets accumulated in the brain, it is responsible for mental retardations. So because of this accumulation in the brain, it causes mental retardation. So one defect is mental retardation. Then next one, because phenylalanine is not converted to tyrosine, tyrosine will not be converted to melanin. Since melanin is not there, hair will not have any color. Now eyeball will not have any color, complexion, body color will not be any there. So this we call it as an albinism. No color for the body, no color for the eyeball, no color for the hair. It's called as albinism, albinos. This is the second change what we can see. And next one, phenylalanine. It's a protein metabolism disorder, right? Phenylalanine is an amino acid. So this protein metabolism disorder, what it will do is it converts phenylalanine into ketone bodies, and those ketone bodies gets excreted in urine. That's why the disorder is called phenylketonuria. Protein metabolism gets disturbed and then ketone bodies are formed, and those ketone bodies are excreted in urine. That's why the disorder is phenylketonuria. The third one we can tell, ketone bodies are excreted in urine. Ketone bodies are excreted in urine. So one gene getting mutated, one enzyme defect is showing so many changes. Mental retardation, albinism, ketone bodies accumulation, all this, reduced IQ, all this are examples for one gene change. So can we call it as Pleiotropy, plural, many changes. So that's why we take phenylalanine. Phenylketonuria is an example of Pleiotropy. We can also take one more example of Pleiotropy, which is called as sickle cell anemia. We can also take sickle cell anemia as an example. Sickle cell anemia, what happens is also an example of point mutation. So in sickle cell anemia, because of the change in one particular gene or one particular codon, what happens means the normal shape of RBC changes to sickle shape. So if it changes to sickle shape, then its oxygen transporting ability will decrease, breathing rate will decrease, heart rate will decrease, the lifespan of RBC is reduced. So many changes we are talking, and they are resistant to malaria. So many changes we are taking because of the defect of one gene. So that's why sickle cell anemia is also example of Pleiotropy, and phenylketonuria is also example of Pleiotropy. Take a screenshot, then we'll get to the next topic called sex determination. Right? So the next topic is sex determination. So the mechanism of sex determination has always been a puzzle for geneticists, and due to the investigations of some scientists, they have narrowed and they came to a conclusion that chromosomes are the basis of sex determination. So the next topic is sex determination. So the Ncert, the author states that mechanism of sex determination has always been a puzzle before Genesis. The initial clues of genetic or chromosomal mechanisms of sex determination can be traced out by the experiments carried by scientists in insects. So we have narrowed our search that what is responsible for sex determination means it's a genetic or the chromosomal mechanism. So the initial clues the initial clues about genetic or chromosomal basis of sex determination came from the experiments carried in insects. Now, who are the scientists who started working on this concept? Means there's a scientist called Henking in 1891. So the first scientist is Henking. In the year 1891, he started working on spermatogenesis in the insect called wasp. He is observing spermatogenesis in an insect called wasp. Now what he saw is 50% of the sperms. 50% of the sperms. He observed that they are receiving a specific genetic structure during spermatogenesis, while the remaining 50% of sperms are not receiving this specific genetic structure. This was the conclusion, this was the observation initially done by scientist Henking in the year 1891. He was observing the process of spermatogenesis in an insect called wasp, and he saw that 50% of the sperms are receiving a specific nuclear structure and the other 50 are not receiving that. So he gave the name to that specific nuclear structure as X body. So he gave the name to the specific nuclear structure as X body, but he did not give the significance of that. He did not give the significance of that. He did not tell that's involved in sex determination. Later, another scientist, his name is Klung. Later, another scientist came. His name is Klung. He also observed spermatogenesis in insect, but not in wasp. He observed it in grasshopper. Later, Klung observed spermatogenesis process in another insect called grasshopper, and then he told that this X body, what Henking has told, is responsible for sex determination, and he renamed this X body to X chromosome because it is responsible for the sex of an individual. Later, Klung, he observed spermatogenesis in grasshopper, and he reported that the X body is nothing but the X chromosome, and he gave the name X chromosome. He told the X body by Henking is nothing but X chromosome. So he is a person who coined the term X chromosome, and he told X chromosome is responsible for sex determination in grasshopper. Is responsible for sex determination in grasshopper. This type of sex determination comes under X0 type of sex determination. It comes under X0 type of sex determination. Take a screenshot of this. We will continue. What did we discuss here? We are talking about sex determination. So it's a puzzle for the scientists or geneticists, like what is the basis of sex determination. The initial clues came from the genetic and chromosomal basis. When they are conducting experiments on insects, they concluded that it is the genetic basis which is responsible for sex determination, or chromosomes which are responsible for sex determination. The initial discovery by Henking in 1891, he was observing spermatogenesis. He told 50% of the sperms will receive something, some nuclear structure. The other 50% sperms will not receive. So he called that nuclear structure as X body, but he did not give it significance. Whereas Klung conducted the same experiments in grasshopper, and he told this is the one which is responsible for sex determination, and since it is responsible for sex determination, he called it as an X chromosome. Take it, we will continue further. Right. So there are different types of sex determination. The type of sex determination what we are discussing is X0 type of sex determination. Let's go with the first type of sex determination. It is X0 type of sex determination. So where do we find the sex determination? In large group of insects. It's found in large group of insects. Like grasshopper, they observed first. Now here, if you see the female insect and the male insect. The female insect along with the autosomes, it bears an additional X chromosome. So it has one X chromosome and an additional X chromosome, whereas the male insect along with the autosomes will have only one X chromosome. The male insect along with the autosome will have only one X chromosome and the female will have XX chromosome. That's why we call the sex determination as X and 0. Only one X is there, additional X is not there. So X0 type of sex determination. So the female has XX condition, the male will have X0 condition. This is the male symbol and this is the female symbol. Now during the formation of X, the female will make eggs with X chromosome. The male will make two types of sperms. One sperm which is having this X chromosome, another sperm which is not having the X chromosome, 0, not having the X chromosome. Now when they get fertilized, this X with this sperm, it will be X and X. This egg with this sperm, it will be X0. Parallelly this egg with this sperm, XX. Parallelly this with this one, X0. Right? XX will turn to be female insects. XX female insects. X0 male. X0 male. Here who is responsible for sex determination? The sperm is responsible for sex determination, because the sperm, if it is not bringing the X body, they are becoming males. The sperm, if it is bringing the X body, it's becoming females. So this is who are heterogametic here. The males are heterogametic here. The males are heterogametic. Heterogametic means males are producing two types of sperms, whereas females are homogametic, because female will produce same types of eggs only. This type of sex determination is called as X0 type of sex determination, which is seen in large group of insects. Example is grasshopper. Take a screenshot, then we will talk about the other type of sex determination. Yes, the second type, in this sex determination, the males are having one chromosome lesser than the females, right? The males are having one chromosome lesser than the females. So one important point is the males are having one chromosome less than females. Obviously autosomes we wrote same here, but here they are having 2X, but here they are having X and 0. So the males will have one chromosome lesser than the females, right? The number of male chromosomes are less. So now let's go to the second type of sex determination, which is called as Z0 type of sex determination. This determination is seen in insects and grasshoppers. This sex determination is seen in butterflies and moths like Fumia. These are the examples. Now here we need to tell that the females will have Z0 condition and the males will have ZZ condition. The chromosomes, we can call it as X chromosome, Y chromosome. We can also take the other chromosomes as Z and W chromosomes. So here the females are Z0 and the males are ZZ. That means the males here will produce same type of sperms, Z and Z, whereas the females will produce two types of eggs. one egg with Z chromosome, the other egg without Z chromosome, 0. Then what type of offsprings we get? When the egg fuses with this sperm, we get ZZ. When the egg fuses with this sperm, also we get ZZ. If this egg fuses with this one, we get Z0, and if this egg fuses with this sperm, also we get Z0. ZZ means male. This is male. Again, ZZ means male, whereas Z0 is female. Z0 is female. Now, who are responsible for sex determination? It is the egg which is bringing Z or 0. If it brings 0, it is becoming female. If it is becoming Z, it is becoming male. That means in this sex determination, females are heterogametic. Females are heterogametic. They are producing two types of eggs, whereas the males are homogametic. So this is second type of sex determination, that is ZZ and Z0 type of sex determination seen in butterflies and moths like Fumia. Take a screenshot. We'll get into the third type of sex determination next. Now the third type of sex determination is XX and XY type of sex determination. The third type of sex determination is XX and XY type of sex determination. Now this is seen in humans and Drosophila, fruit fly. XX and XY type of sex determination is seen in humans and Drosophila. Here the females will bear the allosomes XX and the males will bear the allosomes XY. The chromosomes which are common in both males and females are called autosomes. The chromosomes which are dissimilar in males and females are called allosomes. So X, Y, Z, W are allosomes. A and A, if we write, means they are autosomes. Now in XX and XY sex determination, females have XX allosomes, males will have XY allosomes. So females will produce only eggs which are having X only, whereas the males will produce two types of sperms, one sperm with having X chromosome, the other sperm which is bearing Y chromosome. Now when the egg and sperm fuses, if X fuses with X, then if the X fuses with Y, what is the condition? If this egg fuses with this sperm, XX. If this egg fuses with this sperm, it is X and Y. XX is female. XY is male. XX is female, XY is male. So here the sperm is deciding, because egg is bringing only X chromosome for girl and for boy both, whereas if the sperm brings X, it becomes female. If the sperm brings Y, it becomes male. That means in XX, XY type of sex determination, males are heterogametic, right? Males are heterogametic because they are producing two types of sperms, whereas females are homogametic. Females are homogametic. Understood? Take a screenshot. We'll talk about the next type of sex determination. Right. The fourth type of sex determination is ZZ and ZW type of sex determination. The fourth type of sex determination is ZZ and ZW type of sex determination. This sex determination is seen in birds like chicks and reptiles. This sex determination is seen in birds and reptiles. Now, here when we see ZZ is a condition going with male and ZW for female. So the female bears the allosomes called ZW and the males will bear the chromosomes called ZZ. So here males are homogametic. So here the males are homogametic. Means they can produce same type of sperms. A sperm which is bearing Z chromosome a sperm which is bearing Z chromosome, but the female can produce two types of eggs, an egg with Z chromosome, an egg with W chromosome. Now if we see the combination, if this egg fuses with this sperm, it will be ZZ. If this egg fuses with this sperm, then also ZZ. If this egg with this sperm, ZW. If this egg with this sperm, ZW. ZZ is male. ZZ is male. ZW is female. ZW is female. So appearance of W is deciding the sex here. If two Z's are there, then it is male. If Z and W is there, it is female. So that means females are heterogametic because they're making two types of eggs here. Males are homogametic. So females are heterogametic. So this is the fourth type of sex determination. The first type is X6, X0. So it was seen in large number of insects like grasshopper. Next one is ZZ, Z0, seen in butterflies, moths and fumia. In these two sex determinations, so they have one chromosome lesser than the normal one, whereas X6 and XY, they're having equal number of allosomes. ZZ and ZW also having equal number of X chromosomes. X6 and XY is seen in humans and Drosophila. ZZ and ZW is seen in birds and reptiles. Take a screenshot, then we will talk about sex determination in honeybees. Now in honeybees, the sex determination is called as haplo-diploid method of sex determination. In honeybees, the sex determination is called as haplo-diploid method of sex determination. Now, what is this sex determination? Let's try to understand carefully. Now, here female bees will be there and male bees will be there. The males are called drones, right? Now the females are diploid, it seems. The males are haploid, means males are having 16 chromosomes. 16 twos are 32, the females will have 32 chromosomes. Now if female is having 2N condition, when the female is undergoing gametogenesis, oogenesis, then they are going to do meiotic division. Because it is 2N, you can do meiotic division. Then she will produce eggs which are haploid. The eggs are haploid, means having 16 chromosomes and having 16 chromosomes. But since males are haploid only, they cannot undergo meiosis, they do mitosis for spermatogenesis. Since males are haploid, during spermatogenesis, they cannot do meiotic division, reduction division, they will do mitotic division and they make the sperms which are having 16 chromosomes. Now, if the eggs are fused with this sperm, if sperm and egg are fused, if the sperm and egg are fused, so 16 plus 16, you will get 32 chromosomes. 32 chromosomes is diploid number, right? 32 chromosomes is diploid number. Diploid number means it is female. It is female. And if the egg is not fertilized, if the egg is unfertilized, if the egg is unfertilized, and unfertilized egg if directly develops into an embryo, if it directly develops into an young one, the process of development of embryo without fertilization is called as parthenogenesis. So that process where unfertilized egg directly turns into embryo and young one is called as parthenogenesis. The process is called parthenogenesis. By the process of parthenogenesis, here males are coming because it is haploid, egg is haploid. 16 means it is haploid, means male. The process of parthenogenesis where males are obtained is called as arhenotoky. By the process of parthenogenesis, if the males are obtained, then the process is called as arhenotoky. So unfertilized eggs will directly develop into males. unfertilized eggs will develop into males, whereas fertilized eggs will develop into females. Fertilized eggs will develop into females. Eggs which are unfertilized will develop into males. That is why the males are haploid and the females are diploid. Again, in this fertilized eggs, the females which feed on royal jelly will turn into queens, and the females which feed on normal nectar and honey will turn into worker bees, right? They turn into worker bees. So worker bees are called nurse bees, right? They are sterile. They are not fertile, they are sterile. So the queen is the one which feeds on royal jelly. The fertilized female which feeds on royal jelly will become queen, and the fertilized female which feeds on honey will turn into a worker bee, will turn into nurse bee, and they are sterile. Means here the worker bees cannot lay eggs, they are sterile. The queen only can lay the eggs. The queen will lay the eggs by meiotic division, whereas the males who are haploid will lay the sperms by mitotic division only. So this type of sex determination, where haploid condition is seen in males and diploid condition is seen in females, is called as haplo-diploid method of sex determination. So this is about sex determination. Take a screenshot, then we'll talk about the next concept, mutation. The next topic is mutations. So what are mutations? A sudden change in the DNA sequence, in the gene sequence, is called as mutation. And who is called as father of mutations? Hugo de Vries is called as father of mutations, right? So yeah, Hugo de Vries is father of mutations. He worked on mutational studies, so that's why he is considered as father of mutations. On which plant did he conducted mutations experiments? In a plant called evening primrose. Its scientific name is Ornithera lamarckiana. He conducted mutation experiments. So what is mutation? Mutation is sudden change in the genetic material. It's a phenomenon which results in alternation. It results in changes in the DNA sequence. If there's a change in DNA sequence, then the genotype will be changed as well as the phenotype will be changed, which results in the change in the genotype and phenotype of an organism. Now variations are because of recombinations also, are because of mutations also. In addition to recombinations, mutations are also the reason for variations in the DNA. And where is DNA located? DNA is located in the chromatid, and in this chromatid DNA runs from this end to the other end, right? So DNA runs from one end of the chromatid to the other end of the chromatid. And where is chromatid present? It's present in a chromosome. Any change, gain or loss of a DNA, this much fragment is lost, or sometimes this much fragment might get added. So gain or loss of a segment of DNA will change the structure of the chromosome also. So that's why we call the chromosomal changes as chromosomal abnormalities. We can also call them as chromosomal aberrations, which are commonly seen in cancerous cells, oncogenic cells, cancerous cells. Mutations can arise due to change in single base pair substitutions also. Mutations can arise due to single base pair changes also. Then at one point A is removed, G is added. So we call it as a point mutation, single mutation. Example, classical example for point mutation is sickle cell anemia. We will study about the mutations in detail in molecular basis of inheritance chapter number 6. There we will study it in detail. Here in principles they have given this much only. And mutations in one base pair is called point mutation. Mutations of addition of bases or deletion of bases, substitutions, is called point mutations. Deletion and additions, insertion of base pairs will cause frame shift mutation. Again, frame shift mutation we'll study it in molecular basis, right? And Hugo de Vries is father of mutation. He worked in a plant called evening primrose, which is called Ornithera lamarckiana. The agent which causes mutation is called mutagen. The mutagen can be physical mutagens are there, chemical mutagens are there. Colchicine, which causes chemical mutation, UV radiations will cause physical mutations. Then coming to the organism which undergoes mutation is called mutant. The organism which undergoes mutation is called mutant. The agent which causes mutation is called mutagen. The process is called mutation. What is mutation? A sudden change, alteration in the genetic sequence or DNA sequence. So this is about mutation, which is relevant to this chapter. Clearly about mutations is there in the next chapter, molecular basis, there we can discuss. And then the next topic is pedigree analysis. Take a screenshot of this. We'll go to the next topic, pedigree analysis. Right. So the next topic is pedigree analysis. So if we want to cross one plant with another plant, so we can easily do it. But in animals or in humans, controlled crosses are not possible. So we cannot cross one person with the other person as of we want, right? So it is not possible to do controlled crosses in animals or in humans. They have designed, the geneticists have designed pedigree charts which will help us to check the possibilities. It's a graphical representation of the probabilities of the genotypes. What is that called? Pedigree analysis. So for drawing the pedigree charts, so we are going to use certain symbols. Now this symbol is used for normal male and circle symbol is used for normal female. If the person is affected, male is affected, so this shows affected male, and this pedigree symbol shows affected female. Now this is mating between father and mother, right? And if the father and mother are near relatives to each other, so two lines we need to say, this is called consanguineous mating. So what is this called? This is called as consanguineous mating. This is consanguineous marriages. Means marrying among the relatives is called as consanguineous marriages, and these are the parents and the offsprings are represented in this manner. So one daughter and one son we represent in this manner. Now if they are twins, then we represent in this manner. So for twins we use this type, and for normal children we use like this. And then if the sex of the child is not specified, then we go like this. If the sex of the child is not specified, if the sex of the child is not specified, we go like this. For twins we use this symbol, for normal children we use this symbol, right? And then next one, if they are carriers, we can show like this also, or we can do half the way also. Both the symbols will denote they are carriers of the disease. It denotes they are the carriers of the disease. Now if they symbolize like this, or if they symbolize like this, they are deceased, they are no more, they are dead. They are dead. So these are the symbols we use for drawing the pedigree charts. Now let us see, what did we discuss? This symbol is for a normal male, healthy male. This is for healthy female. This symbol which is filled is affected male. Filled circle will denote affected female. Now male and female, one line denotes mating, and male and female, two lines will denote that they are relatives, and such a marriage is called consanguineous marriages. Male and female, father and mother and two children, normal will draw like this. If they are twins, we draw like this. If the sex of the child is not specified, then we draw a diagonal symbol like that. Carriers of a disease, they are not affected but they have the defected gene within, they're called carriers. We can use this symbol. For male we use it like this, right? And if the persons are dead, if they are no more, so we use this symbol. So these are the pedigrees. Now we need 2022. So on September last Saturday, right? It is on which date? It's on Sunday, 4th September. They have conducted re-examination. Re-examination 2022 they conducted on September 4th. There was a question on pedigree charts. In that they asked identify the mismatch. So this they have given, which is correct. This they have given, which is correct. This they have given, which is correct. Fourth one, they gave this. They gave this and they told it is normal mating. Is it normal mating or it's consanguineous marriages? It is consanguineous marriages. So this they went it mismatch, right? This is a wrong answer. So you can check the paper, re-NEET examination, which is conducted in 6 places on September 4th. There was a question on pedigree. Take a screenshot, we will continue. We have seen the symbols of pedigree chart. Now let us see the information about pedigree chart, what they have mentioned in the Ncert textbook. Now, as I told you, controlled crosses, we can do it in P experiment. Mendel has done it. But controlled crosses in humans, it is not possible, right? So, since controlled crosses as conducted in P plants are not possible for few humans, study of family history about the inheritance of a particular trait. So, you're taking a particular character or trait and you are studying that trait, how does that appear in a human family? Provides an alternative. It provides an alternative. Such an analysis which you are doing in a family for a particular genetic trait is called as pedigree analysis. Now, in a pedigree analysis, we are going to look at the inheritance of a particular trait, right? And we are going to look at a family tree over the generations. That's about pedigree analysis. In humans, pedigree analysis provides a strong tool which is used in tracing out a disorder or abnormality or a disease. We can find out genetic disorders by using pedigree analysis. Now, what is genetic disorder? Genetic disorders are due to changes in the gene sequence or changes in the chromosomal sequence. So, genetic disorders are classified into two types: gene disorders and chromosomal disorders. Gene disorders means changes in a gene, changes in a DNA sequence is called as gene disorder, which can also be called as Mendelian disorder. Mendelian disorders means they are transmitted from one generation to other generation. That's why Mendel worked on inheritance. So that's why, since these genetic disorders are transmitted by the process of inheritance, we call gene disorders, DNA disorders, as Mendelian disorders, and Mendelian disorders are inherited. Whereas disorders on the chromosomes, chromosomal abnormalities, addition of chromosomes, deletion of chromosomes, right? It comes under chromosomal disorders. So they are not transmitted. So that's why chromosomal disorders are called as non-Mendelian disorders. Now we know that the chromosomes which are common in both male and females are called autosomes. The chromosomes which will help in sex determination, which are unique for male and which are unique for female, are called as allosomes. Now there is a disorder in a gene or in a DNA sequence. Where is it present? It's present in the autosome or it's present in the allosome. So on the basis of that, we can divide the Mendelian disorders into autosomal disorders and allosomal disorders. Again, in autosomes, is it a dominant allele or a recessive allele mutation? So we can classify autosomal dominant and autosomal recessive. In the same manner, coming to allosomes, allosomes are X and Y chromosomes in humans. So whether it is X dominant or X recessive. Why? Since only one Y is there, we cannot call this dominant or recessive, we call it a Y-linked disorder. Under allosomal, we have X dominant disorder, X recessive disorder, and Y-linked disorder. Now coming to chromosomal disorders also, in chromosomes also we have autosomes and allosomes. So chromosomal disorders, non-Mendelian disorders, are again divided into autosomal and allosomal. Now actually we are diploid, we have two chromosomes. This condition, every chromosome having two each, is called as disomy. But due to some changes in cell division, sometimes only one chromosome is inherited. Instead of two, only one chromosome is inherited. That condition is called monosomy. Monosomy means missing of one autosome. Autosomal monosomy. Sometimes instead of two chromosomes, because of abnormalities in cell division, three such chromosomes will come. 21st chromosomes, instead of two, three appeared. So that's called 21st chromosomal trisomy. So trisomy of autosomes, monosomy of autosomes. Similarly, trisomy of allosomes, monosomy of allosomes. So all these are the types of genetic disorders, and if you want, we can write the examples of these genetic disorders also, which we have to see one by one in detail. Now coming to autosomal dominant disorder, the example is muscular dystrophy. What is the example for autosomal dominant? It is muscular dystrophy. Example for autosomal dominant is muscular dystrophy. I'm sorry, it is myotonic dystrophy. Myotonic dystrophy is an example for autosomal dominant. It is a type of muscular dystrophy only. Coming to allosomal recessive, the examples of allosomal recessive are, we can take sickle cell anemia, hemophilia. Examples are sickle cell anemia, thalassemia, sickle cell anemia, then next one is thalassemia, phenylketonuria, cystic fibrosis are examples of autosomal recessive. So memorize the examples of each disorder carefully. Examples for autosomal dominant are myotonic dystrophy. Examples for autosomal recessive are sickle cell anemia, thalassemia, phenylketonuria and cystic fibrosis. In Ncert textbook they have given examples for X-linked recessive. The examples for X-linked recessive are color blindness and hemophilia. Examples for X-linked recessive are color blindness and hemophilia are examples of X-linked recessive. Y-linked recessive, examples for Y-linked recessive are porcupine male and hair on the body and porcupine male. So these are some disorders which are Y-linked disorders. Coming to chromosomal disorders. In chromosomal disorders in NCERT we have three as examples. One is autosomal trisomy, other one is allosomal trisomy, other one is allosomal monosomy. There are three disorders we have to study in detail about them. So in NCERT they have given example for autosomal trisomy. It is 21st chromosomal trisomy which is called as Down syndrome. And for allosomal monosomy, they have given Turner syndrome as an example. They have given Turner syndrome as an example. For allosomal trisomy, they have given Klinefelter syndrome as an example. Klinefelter syndrome is an example of allosomal trisomy. Turner syndrome is an example of allosomal monosomy, and Down syndrome is an example of autosomal trisomy. For Y-linked disorders, hair on the body, porcupine male are the examples. For X-linked recessive disorders, color blindness and hemophilia are examples. And for autosomal recessive, sickle cell anemia, thalassemia, phenylketonuria, cystic fibrosis are the examples. For autosomal dominant, myotonic dystrophy is an example. So since Mendelian disorders are inherited, Mendelian disorders can be traced out with the help of pedigree charts, which runs in families. By drawing the pedigree we can check the probability. Whereas the chromosomal disorders, since they are not inherited, so that's why we cannot trace the chromosomal disorders through pedigree analysis. Take a screenshot, then we will see one by one disorder in detail. We are talking about gene disorders. Gene disorders are also called Mendelian disorders. They can be inherited and they can be predicted to pedigree diagrams also. So gene disorders are of two types. If the disorder gene is on the autosome, then that is called autosomal disorder. If the disorder gene is on the allosome, that's called allosomal disorder. Again, if the gene is dominant, then that's called autosomal dominant disorder. If the gene is there in the autosome and if it is a recessive gene, it is called autosomal recessive. Now let us see one by one. So I have written the content on the board. Let's go through. Under autosomal dominant, there's only one example which is mentioned in NCERT, that is myotonic dystrophy. There's one disorder which comes under autosomal dominant. What is that? It is called myotonic dystrophy. In NEET 2022 examination, which happened in the month of August, right? August 17 they conducted examination. In that paper they asked a question like, which of the following occurs due to the presence of autosomal linked dominant disorder, and they have given sickle cell anemia, hemophilia, myotonic dystrophy and thalassemia as an example. So they asked you autosomal dominant disorder. Only one example for autosomal dominant disorder which is mentioned in NCERT is myotonic dystrophy. So the answer is myotonic dystrophy. So I am telling you that this disorder has been asked in NEET 2022 examination. Now let's quickly see what is myotonic dystrophy. As we know that myotonic dystrophy comes under autosomal dominant disorder. Means it is present on autosome. How many autosomes do we have? 22 pairs of autosomes we have. This particular gene is located on the long arm of chromosome number 19. Long arm means Q segment, right? It is on the Q segment of the 19th chromosome. That gene codes for an enzyme which is called as myotonic dystrophy protein kinase. There's a gene present on the long arm of the 19th chromosome which codes for an enzyme called myotonic dystrophy PK protein kinase enzyme. Because the gene is disordered gene, protein kinase will not be made. Since protein kinase is not made, what happens? There is a progressive weakness in the skeletal muscle. Due to this gene disorder, enzyme production stops. When the enzyme production stops, then there is a progressive degeneration of skeletal muscles, not smooth muscles or visceral muscles. Skeletal muscles progressively they get degraded, weakened. That disorder is called myotonic dystrophy. I think we understood what is meant by myotonic dystrophy. It comes under autosomal dominant. The gene is located in chromosome number 19. It's on the long arm. Now let's go to autosomal recessive disorders. The disordered gene is present on autosome, but it's not a dominant allele, it is a recessive allele. Now for autosomal recessive, there are many examples. Let us see one by one. First, I have written there cystic fibrosis. That's one example. Second one, thalassemia. That's the second example. Third one, sickle cell anemia. And the fourth one is phenylketonuria. There are 4 examples for autosomal recessive. One is cystic fibrosis. Now, cystic fibrosis is autosomal recessive. On which chromosome it is present? It is present on chromosome number 7. So you can remember like F is like 7, right? It is the reverse form of 7, like that you remember. Cystic fibrosis is located on chromosome number 7, right? So there is a gene which is a recessive gene which is present on chromosome number 7. Now because of the defective gene, it codes for a defective glycoprotein. Because of the defective gene, it codes for a defective glycoprotein. This defective glycoprotein makes the mucus thick in the lungs. Because of that thick mucus in the lungs, cystic fibrous cysts appear in the pancreas, and sodium transport, potassium transport gets interfered. Because of that, the disorder is caused, which is called cystic fibrosis. So we are talking about autosomal recessive disorders. The first example is cystic fibrosis. The defective gene is located on chromosome number 7. It codes for a defective glycoprotein which makes the mucus thicker. When it becomes thicker, that mucus deposits in the lungs and it forms fibrous cysts in the pancreas. It interferes with the mineral transport. That's it. Let's come to the second disorder which is under autosomal recessive, that is thalassemia. Thalassemia, where is the gene located? There are 2 types of thalassemia. Alpha thalassemia, beta thalassemia. Alpha thalassaemic gene is located on chromosome number 16. Beta thalassaemic gene is located on chromosome number 11. Now what is thalassemia? Thalassemia, the gene got defected because of deletion or because of mutation. That's what they mention in NCERT. Thalassemia is a defect. It could be due to deletion or it could be due to mutation. So when there is deletion or mutation of a gene, then there is defective in the synthesis of globin. Hemoglobin is made up of a pigment called heme and it's made up of protein called globin. Now there are 4 globin chains, right? 2 alpha chains and 2 beta chains. Hemoglobin contains 4 polypeptides. 2 polypeptides are alpha, 2 polypeptides are beta. Now if the alpha polypeptide chain synthesis is affected, it's alpha thalassemia. If the beta polypeptide chain synthesis is affected, it is beta thalassemia. Now because of that, what happens? Alpha chains are not synthesized, so the number of hemoglobin molecules gets reduced, means it is quantitative disorder. The number of hemoglobin molecule, if it gets reduced, it leads to anemia. It leads to anemia. So here I wrote thalassemia as 2 types, alpha thalassemia and beta thalassemia. As I told you, in alpha thalassemia, the alpha chain synthesis gets affected. Now, where is alpha thalassemia gene located? I told you it is located on chromosome number 16. Now, there are two genes which code for alpha thalassemia, HbA1 and HbA2. So, we know that each gene has two alleles, right? So, HbA1 has two alleles, HbA2 has two alleles, two genes and four forms. If one allele is affected, then the occurrence of the disease will be less. If two alleles are affected, then more hemoglobin molecules get affected. If 3 and 4 get affected, then many hemoglobin molecules gets affected. So more the genes affected, then lesser the alpha chains are made. More the genes affected, then more the disorder. When more the disorder, then less alpha chains occur. So because of that, anemia will happen. Now coming to beta thalassemia, I told in beta thalassemia, beta polypeptide chain synthesis gets affected. And the gene is located on chromosome number 11. And it is a single gene. Because it is a single gene, we will call HBB. A gene has two alleles. Means HBB gene, one allele can get affected or both the alleles get affected. If both the alleles are affected, the disorder is severe, right? So mutation of one or both the alleles or genes in the beta chain affects the synthesis of beta chain. That's about thalassemia. And I told you it comes under qualitative disorder or qualitative disorder. Here it is a quantitative disorder. The number of hemoglobin molecules are reduced, leading to anemia. So it comes under quantitative disorder because the number of chain synthesis gets affected here. Take a screenshot, then we will get into the third disorder, which is called sickle cell anemia, and the fourth disorder called phenylketonuria. Take a screenshot of this. Right. Now let's talk about the third type of disorder, which is autosomal recessive only. It's called as sickle cell anemia. This part of principles of inheritance and variation, where the genetic disorders will come, pedigrees will come, is a very important concept. And as I told you, myotonic dystrophy question has been asked in NEET 2022 examination. There are 3 questions which they asked in NEET 2022 examination. We'll see when it comes. On hemophilia they asked one question, on thalassemia they asked one question, on myotonic dystrophy they asked one question. No, right? We are talking about autosomal recessive disorders. We completed 2 examples. The first example is cystic fibrosis. The gene is located on chromosome number 7. The second one we just now completed, thalassemia. For alpha, it is located on chromosome number 16. Beta, it is located on chromosome number 11. Now coming to the third example, which is under autosomal recessive, which is very important, is called as sickle cell anemia. So the third disorder, what is the third disorder called? It is called sickle cell anemia. Now the gene which codes for sickle cell anemia, where it is located, it's an autosomal recessive. It is located on chromosome number 11. The third disorder for autosomal recessive is sickle cell anemia. Now sickle cell anemia, first point, it is autosomal recessive disorder. Where is the gene located? The gene is located on chromosome number 11. So where is the gene located? Gene is located on chromosome number 11, right? Now there are 3 possibilities. That the 3 genotypes are, out of the 3 possible genotypes, let's write down. Let us see what are the 3 possible genotypes. One genotype will be HbA and HbA. HbA is adult hemoglobin, means this person is normal individual. The other possibility will be carrier individual. One is HbA, which is a normal hemoglobin. The other one is sickle cell hemoglobin. This is a recessive one. This is suppressed by this one. So here they are called as carriers. And the third possibility will be HBS and HBS, affected individual. This is the third possibility, affected individual. So in normal hemoglobin, adult hemoglobin, what is the sequence of the DNA? So the DNA sequence on the coding strand, the DNA sequence will be GAG, and on the template strand, the DNA sequence will be CTC. From the template strand, mRNA is made. The codon of this mRNA will be GAG, and this is the sixth codon. This sixth codon codes for the sixth amino acid. We know the genetic code that GAG codes for glutamic amino acid. So glutamic acid is the sixth amino acid. Glutamic acid is the sixth amino acid. Of which chain? Alpha chain or beta chain? Sickle cell anemia is defect due to beta chain. So we need to write the beta chain amino acid sequence. This is the beta chain. In beta chain, the first amino acid we need to write. The first amino acid is valine. The second amino acid is histidine. The third amino acid is leucine. The fourth amino acid is threonine. The fifth amino acid is proline. The sixth amino acid is glutamic acid. The seventh amino acid is also glutamic acid. This is what the sequence what they have given in NCERT textbook. In sickle cell anemia, the alpha chain synthesis gets affected. In alpha chain, the sixth codon undergoes mutation. This is the sixth codon. It undergoes mutation. Which mutation is that? So T got converted to A, right? T, this is a normal thing. This is a normal thing. T codes for A only. This is a normal gene. I'm sorry. This is a normal gene. The first amino acid is valine. The second amino acid is histidine. Third one is leucine. Fourth one is threonine. Fifth one is proline. Sixth is glutamic acid. And seventh one is also glutamic acid. If this is the amino acid, then the hemoglobin molecule will be normal hemoglobin and it will be biconcave in shape. It will be biconcave in shape, right? So when we take the HBS gene, mutated gene, in HBS gene, so the coding strand, we have a sequence called G, it will be G, instead of A, it gets converted to T, GTG. Then here it gets converted to CAC on the template strand. Then this codes for an mRNA. So C codes for G, A codes for U, and C codes for G. So that means the sixth codon, which should be GAG, got converted to GUG. There's a mutation here. Which mutation is this? Point mutation. Mutation at a point, it is point mutation. So sickle cell anemia is a classical example of point mutation, and it is substitution. It's not deletion, it's not insertion. There is substitution of A with U. So it comes under base pair substitution. It comes under base pair substitution. It comes under point mutation. Now this GUG codes for which amino acid? GUG, we know that it codes for valine. The first to fifth amino acids are the same. If we want, we can write here. The first amino acid is valine. Second amino acid is histidine. Third amino acid is leucine. Fourth amino acid is threonine. Fifth amino acid is proline. The sixth one will be valine and seventh one will be glutamic acid. Which chain is this? This is beta chain of hemoglobin molecule. Because of this sickle cell chain, what happens? It's not normal hemoglobin. It becomes a sickle cell hemoglobin. It becomes a sickle cell hemoglobin. So this is that's why it's called as a genetic disorder, because there's a defect in the gene. Take a screenshot. We will continue. Right. So when we see here, these persons are normal, they code for normal hemoglobin. These persons are carriers. Now carrier can transmit the disease, right? They have 50% chances to transmit the disease, whereas affected individuals will not survive usually because they have very less amount of hemoglobin. So that's why they can't survive. HbA people will have normal hemoglobin, whereas HbA and HbS people who are called carriers, they will also have normal hemoglobin at normal oxygen concentrations because HbA is dominant. But when the oxygen tension goes low, when the oxygen tension goes low, when we are climbing higher altitudes like that, when the oxygen tension goes low, at that condition HbS gets polymerized and it makes the situation even worse. So when the oxygen tensions are low, then HbS gene, the defective gene, undergoes polymerization. It undergoes polymerization and converts biconcave RBC to sickle shape RBC. It converts biconcave RBC to sickle shape RBC and it makes the situation worse, right? So this is about sickle cell anemia. So we can take sickle cell anemia as an example of autosomal recessive disorder. This is one example. Its example is a classical example of point mutation. Which type of point mutation? It is substitution. And which got substituted? Like A got substituted to U, right? So means a purine got substituted to pyrimidine. So it's called transversion. It's called transversion and it is an example of Pleiotropy. Sickle cell anemia is also example of Pleiotropy. What do you mean by Pleiotropy? One gene showing many defects, right? One gene showing many phenotypic manifestations is called Pleiotropy. What are the many manifestations? What are the many changes we can see? The first change is shape of RBC is changed. Shape of RBC is changed. Lifespan of sickle cell RBC is low. The lifespan is changed. Oxygen carrying ability will decrease. Oxygen carrying ability is decreased. Respiratory rate is decreased. Circulation is decreased, right? And then malarial parasite Plasmodium can infect the RBC if it is like this, but if the RBC gets converted to sickle cell, it can't identify it as RBC. Means people who are sickle cell anemia are resistant to malaria. So these people are resistant to malaria. So because they are resistant to malaria, this defective gene is there in these people as a genetic load. It comes in evolution. So sickle cell anemia can also be an example of genetic load. So all these are the terms which are important related to sickle cell anemia. Before we discussed about thalassemia, I told thalassemia is a quantitative problem. Here the alpha chain synthesis get affected and the beta chain synthesis gets affected. Means thalassemia is quantitative disorder. Quantity is affected here. Whereas this is a qualitative disorder. RBCs are made but the quality of RBC is different. So sickle cell anemia can also be an example of qualitative disorder. Sickle cell anemia can also be an example of qualitative disorder. Take a screenshot. We will continue with the next disorder, phenylketonuria. Right. The fourth example, this is the third example. The fourth example of autosomal recessive is phenylketonuria, which is an inborn error of metabolism. It's an enzyme disorder. That's why we call it as an inborn error of metabolism. Third example just now we saw sickle cell anemia. It's on chromosome number 11. Coming to the fourth disorder, it is called phenylketonuria. Now, phenylketonuria, it comes under inborn error of metabolism. This is also an example of autosomal recessive disorder. The affected gene is located on chromosome number 12. Phenylketonuria, the affected gene is located on chromosome number 12 in phenylketonuria. Now, what happens in phenylketonuria? In phenylketonuria, because of that affected gene, an enzyme is not synthesized. What is that enzyme which is not synthesized? So, there is no synthesis of an enzyme called phenylalanine hydroxylase. When this enzyme is not synthesized, then phenylalanine cannot be converted to tyrosine. This is one change. If phenylalanine is not converted to tyrosine, then phenylalanine accumulates in brain and it causes mental retardation, right? And then since phenylalanine is not converted to tyrosine, tyrosine cannot be converted to melanin, means no melanin formation, right? That means that is hypopigmentation. It leads to hypopigmentation, which is called as albinism. Hair is not colored, body is not colored, eyeball is not colored. This is called as albinism, right? And then phenylalanine mental retardation will happen, melanin will not be formed, and because of its poor absorption by the kidney, because of poor absorption by kidney, phenylalanine excretes in urine. It excretes in urine as ketone bodies. That's why it is called as phenylketonuria. One gene defect is showing again many manifestations, so that's why we can call phenylketonuria as also an example of pleiotropy. Phenylketonuria is also an example of pleiotropy. So these are the examples of autosomal recessive. First one is cystic fibrosis, second one is thalassemia, third one is sickle cell anemia, and fourth one is phenylketonuria. Take a screenshot, then we will see the pedigree charts for autosomal dominant and autosomal recessive. Right. How to identify whether the pedigree is an autosomal dominant or autosomal recessive? I'll help you. I'll tell you how to identify the disorders. Now, in NCERT they have given two pedigrees. The first pedigree, it's like this. Affected female they have given, normal female, affected male, normal male, and affected female. And the progeny of this parents, this family, 2 daughters normal and 1 son also normal. This affected female, she married a normal man, and the progeny they showed in this manner. How will you identify which type of pedigree is this? Let me draw the other pedigree also which they have given. One son, affected son, daughter, affected daughter, then affected son. Right, so this is the pedigree. We need to identify which type of pedigrees are there. Now see, if you see that the mother is affected here and the mother has transmitted the disorder to boy and girl. She has equally transmitted the disorder to daughter and son, means it is autosomal, right? So if it is, you concluded that it is autosomal because mother has given it to both boy and girl, right? It is autosomal. Then we need to tell it is autosomal dominant or autosomal recessive. Now in the parental generation, affected individuals are there. In the next generation, also affected individuals are there. In the next generation, these three and these three come under one generation. In the next generation, also affected individuals are there. Means, if affected individuals are there in every generation, then consider it as autosomal dominant. And if the mother transmits to son and daughter equally, then also it is autosomal disorder. So that means we concluded this pedigree as autosomal dominant, right? So this is example for this is example of autosomal dominant pedigree. Example for autosomal dominant is myotonic dystrophy. And coming to the other pedigree, here if you see, the parents are not affected. The parents are not affected, but the children are affected. Means that the parents are carriers here. The parents are carriers here, both the parents are carriers and both the parents are equally given for son and daughter, right? So that means equally son and daughter got affected. Again it is autosomal. And there is skipping of generation. If there is skipping of generation, what do you mean by skipping of generation? Means in parental generation people are not affected, in the next generation people are affected. Here they are not affected, here they are affected. If there is skipping of generation, then that is recessive. If girls and boys are equally affected, it is autosomal. So that means this pedigree is autosomal recessive. This pedigree is autosomal recessive. Examples for autosomal recessive, this pedigree can be cystic fibrosis pedigree or thalasssemic pedigree or phenylketonuria pedigree, or it can be sickle cell anemia pedigree. So I think now you got clarity, like if they have given you a pedigree, how to identify whether it's autosomal dominant or autosomal recessive. We will see in the next one whether it's allosomal dominant or allosomal recessive pedigrees also. We will see. Take a screenshot. We will continue. Next, coming to allosomal disorders. Allosomes means X chromosome and Y chromosome. So there are X chromosomal disorders and Y chromosomal disorders. Y chromosomal disorders are not mentioned in NCERT. Under X chromosomal disorders, we have X dominant disorders and X recessive disorders. X-linked dominant disorders, examples are incontinentia pigmenti is one example. Double cortex syndrome is another example. Incontinentia pigmenti and double cortex syndrome are examples of X-linked dominant disorders. These are not mentioned in NCERT. Coming to X-linked recessive disorders, there are three examples which are examples of X-linked disorders. The first one is DMD, Duchenne's muscular dystrophy. Autosomal dominant is myotonic dystrophy. Here it is Duchenne's muscular dystrophy is allosomal X-linked recessive. The second example is hemophilia. The third example is color blindness. These three we are going to see here now. Now coming to X-linked disorder, as we discussed, the first one is DMD, Duchenne's muscular dystrophy. It is an Lethal means it leads to death. It's a lethal recessive disorder. Now there is a gene called DMD gene, Duchenne's muscular dystrophy gene, which is the largest gene which comes in Human Genome Project. And the salient features they have mentioned, the DMD, the normal size of a gene is 5,000 base pairs, but DMD, which is the largest gene which is present on the X chromosome, it has 2,400 kilo base pairs, they will tell. Duchenne's muscular dystrophin gene is present on the, it's the largest gene which is having 2,400 kilo base pairs, located on the short arm of X chromosome. It is located on the short arm of X chromosome. Because of this, DMD gene gets affected in X-linked recessive disorders. So in affected individuals, dystrophin protein is not made in the skeletal muscles, because of which it leads to deterioration of the muscles, and the person will die at a young age, at 15 to 16 years. That's why we call it as a lethal mutation. I think we understood. We are talking about the first X-linked recessive disorder, that is Duchenne's muscular dystrophy. Duchenne's muscular dystrophy, there is a protein. What is that? DMD. Duchenne's muscular dystrophy protein is coded by DMD gene. It's the biggest gene, made up of 2,400 kilo base pairs. It is located on the short arm of the X chromosome. Since the enzyme is not made, then the skeletal muscles leads to deterioration, and the person dies at an young age. That is why it's called lethal disorder. Coming to the second example, it is hemophilia. Hemophilia is also called bleeder's disease. It's also called royal disease. Hemophilia means persons who have hemophilia in them, a minute cut also, it leads to non-stop bleeding continuously. All the 5 liters of blood which is there in the person's body comes out, and it ultimately leads to death only. That's why it's called as bleeder's disease. We know that in blood clotting is affected by 13 clotting factors. One factor will come and activate the other factor, other comes and activates the next factor, next factor. Such a metabolism is called as cascade metabolism. So in hemophilia, out of 13 factors, only one factor of a cascade gets affected. Because of that protein, the complete blood clotting stops. That's what they wrote here. A single protein, a single clotting factor of the cascade is not synthesized, so then the blood will not clot. A simple cut might result in non-stop bleeding, leads to the death. Right? It was first observed in Queen Victoria. It was first observed in Queen Victoria. Mutation happened in Queen Victoria. She became the carrier. Her X chromosome is having hemophilic gene. She became the carrier, and then she spread it in the royal families of Europe. That's why it's also called as royal disease. Right? Now, the possibility of hemophilic female is extremely rare. If you want to see a hemophilic female, if you want to see a hemophilic female, they're telling it's extremely rare. Why hemophilic female is extremely rare? Means at least the mother should be carrier. The mother should be carrier. She should have one normal X chromosome, other one is hemophilic chromosome. And father should be hemophilic. Father should be hemophilic. If father is hemophilic and mother, if she is carrier, then the daughter will be carrier. Now these persons, males, they have only one X chromosome, and if that X chromosome is hemophilic chromosome, so they are unviable at the later ages. Means fathers who are hemophilic, they cannot survive till the marriage age, so then they cannot transmit it to their daughters. That's what Ncert says. Hemophilic fathers are unviable in the later stages of the life, so they can't reach to the marriageable age, so they cannot transmit this gene to their daughters. But if you want, theoretically we can make questions out of that, but practically this is the scenario, right? So possibility of hemophilic female is extremely rare. Why it's extremely rare? Because her father should be hemophilic, and such fathers will not be there, and mother should be at least a carrier. And hemophilia follows a crisscross inheritance. What do you mean by crisscross inheritance? Crisscross inheritance means from father to grandson via carrier female. From male to male via carrier female, from male to male, from father to grandson via carrier female. This is called crisscross inheritance. X-linked recessive disorders will follow crisscross inheritance. Okay, let us see. We're talking about the second disorder, which is called as hemophilia. It's also called as bleeder's disease because a small cut leads to non-stop bleeding and death. It's also called royal disease because it was first appeared. They saw first it in Queen Victoria family. She became the carrier and thus she transmitted that defective hemophilic X chromosome in her families. That's why it's a common disease in the royal families of Europe. And the daughter becoming hemophilic is extremely rare because father should be hemophilic and mother should be a carrier. And this disorder follows a crisscross inheritance. Crisscross inheritance, male to male via carrier female. Take a screenshot. We will continue with the types of hemophilia. There are two types of hemophilia. Hemophilia A and hemophilia B. Depending upon which blood clotting factor is affected, we can divide them into hemophilia A and hemophilia B. In hemophilia A, blood clotting factor 8 is not synthesized. In hemophilia B, blood clotting factor 9 is not synthesized. Now we are talking about the second disorder, which is hemophilia. Is of two types. First one is hemophilia A, which is the severe type of hemophilia. The second one is hemophilia B, which is a mild type. It is the mild form. In hemophilia A, blood clotting factor 8 is deficient. Blood clotting factor 8 is called as anti-hemophilic factor. What's the name of blood clotting factor 8? It is called as anti-hemophilic factor. When this anti-hemophilic factor is not synthesized, then the cascade mechanism will not happen. It leads to hemophilia A. 80% of the hemophilic people are because of this factor deficiency only. Whereas hemophilic B, mild form, we told 20% of the hemophilic people will be because of hemophilia B. It is because of factor 9 deficiency. Which blood clotting factor? It is blood clotting factor 9 deficiency. Blood clotting factor 9 is called as Christmas factor. 20% of hemophilia belongs to hemophilia B, right? So in NEET 2020 examination, there is a question about hemophilia. So re-NEET exam happened in 2022, right? So when did they conduct it? They conducted it in the month of September 4th. Now in that paper, you can see a question on hemophilia. Let us see what's the question they have asked here. A normal girl whose mother is hemophilic. The question is, a normal girl whose mother is hemophilic marries a man who doesn't have hemophilic history. Then they're asking what will be the possible phenotypes of the offsprings. The normal girl whose mother is hemophilic. So she is a normal girl. Her mother is hemophilic. Right? Then she marries a man. He is normal, means X and Y chromosome. Mother is hemophilic, means XH and XH. Now mother will give 1, that means she is a carrier, means she has XH gene. Since they told normal girl, she has normal X. right? So they are asking what percentage of offsprings will have the disease. 2 sons and 2 daughters. The sons will receive Y chromosome from the father, and mother will give X chromosome and XH. Then next, the daughters will receive this X chromosome from the father. One daughter will receive this X, another daughter will receive this X. That means this son is normal son, affected son, this person. Second son is affected son. This daughter is normal daughter because both are normal X chromosomes. And this daughter is affected daughter. Now the question what they have asked is, what will be the possible phenotypes of an offspring? First option they told hemophilic son and hemophilic daughter. Hemophilic son and hemophilic daughter. Second option they have given hemophilic son and carrier daughter. Yeah, I'm sorry. This is a carrier daughter, right? This is dominant. So she is a carrier daughter. So the second option they have given is hemophilic son and carrier daughter. Third option they have given normal son and normal daughter. Fourth option they have given normal son and hemophilic daughter. Normal son and hemophilic daughter. And they have given 4 options, 1, 2, 3, 4. In the first option they are telling A and B are correct. Second option they are telling B and C are correct. Third option they are telling A and D are correct. Fourth option they are telling B and D are correct. Now if you see, normal son and normal daughter are there. So normal son, normal daughter, C is correct. Affected son and carrier daughter. Affected son and carrier daughter are also there. Means yeah, hemophilic son and carrier daughter, B is there. So check the answer where B and C are there. Second answer means the answer is 2. This is a question when they have given in NEET re-examination. It is on 4th September. I think you understood how to solve this. Take a screenshot. We will get into the third disorder which comes under X-linked recessive, that is color blindness. Right. So under X-linked disorder, the first one we have seen is DMD, Duchenne's muscular dystrophy. The second disorder what we have seen is hemophilia. The third disorder is color blindness. There are 3 types of cones, right, in the retina. Red cone, green cone and blue cone. Red and green color blindness is example of X-linked recessive disorder. So because of the defective cones, they are unable to discriminate between red and green colors. So the third type, red and green cones are defective here. Since it is located on the X chromosome, the occurrence of color blindness in males will be more. So it occurs in males approximately 8%. But in females, the occurrence is 0.4% only. Why it is more in males means because males have only one X chromosome. Since they have only one X chromosome, so the occurrence is more. But in females, since they have 2 X chromosomes, and that too, this is X-linked recessive, the dominant one will subside the recessive gene, so that's why the occurrence in females is just 0.4% only. Now that's why, that means females will be the carriers, males will be affected. Now there's a question on color blindness in NEET 2022 examination which happened on August 17th. Let us see this question about color blindness. The question what they have given is, if a colorblind female marries a male of a colorblind female marries a male. If a colorblind female marries a male whose mother was also colorblind, whose mother was also colorblind, what percentage of the progeny are colorblind is the question. Let's draw the pedigree. If the colorblind female, colorblind mother they told, XC, married a male whose mother is also colorblind. His mother is also colorblind. XC. Then this son will get XC from the mother, XC and Y. So since the son will get XC from the mother and mother is also XC, now when they give birth to 4 children, 2 daughters and 2 sons, sons will get Y chromosome from the father, they will get X chromosome from the mother. This XC will come here, the other XC will come here. Girls will receive this XC from the father for sure, and this XC will come here, this XC will come here. So in boys, if one XC is there, they are affected. Again XC is there, means both the sons are affected. Here both are XC, daughter is also affected. The other daughter is also affected. Now when we draw the pedigree, our conclusion is all the children are affected. So now in the options they have given, first option they have given 25% children are affected. Second option 50% children are affected. Third option 75% children are affected. Fourth option 100% children are affected. We have seen all children are affected, so the answer is 100%. So this is about color blindness, which also comes under autosomal recessive. Take a screenshot, then we will get into chromosomal disorders. Right, so with this we finished gene disorders. The other part is chromosomal disorders. Gene disorders is the defect in a gene, right, which is coding for a protein. But in chromosomal disorder, a part of a chromosome or a complete chromosome is deleted or added. So that's called chromosomal disorder. Chromosomal disorders are called non-Mendelian disorders. They are not inherited, so they cannot be predicted by pedigree charts. So coming to chromosomal disorders, they are also called non-Mendelian disorders and they are not transmitted, so they cannot be predicted by pedigree charts. Now chromosomal disorder, it is due to absence or due to excess of one or more chromosomes. It is due to absence or excess of one or more chromosomes. Now, how does it happen? When does it happen? It is due to failure of segregation of chromosomes during cell division. During anaphase, it leads to addition of chromosome or deletion of chromosome. So, how is this possible? Means it is due to failure of segregation of chromosomes during cell division. In which phase? It is in anaphase 1. Because of failure of segregation of chromosomes in anaphase 1, it leads to addition of chromosome or deletion of chromosomes. Now this is called as aneuploidy. Addition or deletion of chromosomes is called as aneuploidy. aneuploidy. It's called aneuploidy. Now for chromosomal disorders, we have 3 disorders as examples. We can divide the chromosomal disorders into autosomal and allosomal. Under autosomal, again we can tell it is monosomy or we can tell it is trisomy. In allosomal also we can tell monosomy or trisomy. Normal condition is like this, disomy. In monosomy, one chromosome is missing. In trisomy, 1, 2, 3 will be there. That is trisomy. Addition here, deletion here. Monosomy of autosomal, they did not mention. Coming to autosomal trisomy, we have 3 examples. So trisomy of 21st chromosome is called as Down syndrome, and trisomy of 18th chromosome is called as Edward syndrome. Trisomy of 18th chromosome is called as Edward syndrome. Then trisomy of another 13th chromosome is called as Petu syndrome, right? And not a complete chromosome, if a bit of chromosome is deleted, then that is called as Credichat syndrome. In autosomes only, if there is deletion of a part of chromatid of chromosome number 5, then that is called as Credichat syndrome. It's called as Credichat syndrome, which can also be commonly called as cat-cry syndrome. So these many are there, but in NCERT they have mentioned only about Down syndrome. So we will discuss about Down syndrome and then we will go to allosomal. So children, we are talking about allosomal disorders. In allosomal disorders, there is addition of chromosome or there is deletion of chromosome. How is it possible? When it is possible to add a chromosome or delete a chromosome means it is during cell division. Which cell division? Mitosis or meiosis? Meiosis, right? Meiosis, in meiosis 1, in anaphase 1, there is a failure of segregation of chromosomes. So that's called non-disjunction. It is due to non-disjunction of chromosomes, which is called as aneuploidies. We can see addition of autosomes, we can see addition of allosomes. Addition leads to trisomy, deletion leads to monosomy. So take a screenshot, we'll talk about Down syndrome then. The first one is Down syndrome. It is trisomy of 21st chromosome. It's trisomy of 21st chromosome. If we have to write the chromosomal complement, actually we have 46, but here one chromosome is added, it becomes 47 chromosomes. So 44 are autosomes. It's not 44, 45 are autosomes plus X and X, or it can be X or Y, right? XX will be for females, XY will be for males. 45, 46, 47. 45, 46, 47. So trisomy of the 21st chromosome. So in NCERT textbook, they have given karyotype of Down syndrome. If you carefully see, they have given 21st chromosome as 3 in number. Instead of 2, the 21st chromosome, one additional chromosome came. That's why it's called trisomy of the 21st chromosome. That is why instead of 46, they have 47 chromosomes. Autosomes are added, so instead of 44, they have 45, right? It is also called Mongolism. The face appears like a Mongolian face. It is called Mongolism, or it is also called Mongolian idiocy. It's called Down syndrome. It is 21st chromosomal trisomy. It was first observed in a scientist called Langdon Down. It was first reported in a scientist called Langdon Down. What are the symptoms? Symptoms are very important. Let us discuss about the symptoms of Down syndrome. Diagram of Down syndrome child also they have given in NCERT textbook. The first, they are short stature. The back side of the head will be flat. Back head will be flat. The fingers will have many loops. The fingertips will have many loops. Fingertips have many loops. Broad face. Flat face. Big and furrowed and wrinkled tongue. The mouth will not be closed, open mouth, and they have a characteristic palm crease. We have 1, 2, 3 lines here, but for them this line and this line will not be there, so this line will be there and these two lines are merged to form one line. So this is called as characteristic palm crease. So they have a characteristic palm crease and they have congenital heart diseases. By birth itself they have heart diseases. Their IQ will be low, but then they are fertile. And I told chromosomal disorders are not transmitted to the generations. Father or mother, if they are having Down syndrome, they will not transmit it to their next generations. What are the symptoms of Down syndrome? They will be of short stature. Back of the head will be flat. Fingertips will have many loops. Big and furrowed and wrinkled tongue. They can't talk like us fastly. Open mouth, because of long tongue they can't close their mouth properly. Characteristic palm crease will be there. By birth itself they have heart disorders, like heart disorders will be there. IQ will be low, but still they are fertile. Take a screenshot, then we will get into the allosomal disorders. Under allosomal disorders we have allosomal trisomy and allosomal monosomy. For allosomal monosomy, the example is Turner syndrome. So monosomy means one chromosome is missing. Means instead of 46 chromosomes they will have 45 chromosomes, and we told it is allosomal monosomy, means 44 autosomes will be there, but one X chromosome is missing, X0. 44A X0 is the chromosomal complement. So means Turner syndrome is common in females, because one chromosome, X chromosome is there, they are females. Now what are the symptoms of Turner syndrome? The first one, they are sterile. So why they are sterile? Because only one X chromosome is there, right? So hypogonadism, since two allosomes are required, only one X chromosome is there, it leads to hypogonadism. Gonads will not function. Ovaries will not function. Ovaries are rudimentary. They are not completely developed. That's why they are sterile. They don't have neck. The head darkly comes and sits on the base of the body. It's called webbed neck. Short stature. Short stature. IQ will be low. So these are the symptoms of Turner syndrome. So in Turner syndrome, they are sterile, because one X chromosome is missing, hypogonadism, ovaries are rudimentary, menstrual cycle overproduction will not happen, webbed neck will not be there, directly head comes and sits on the body. Then short stature and IQ will be low. Take a screenshot. We'll talk about the last disorder, which is called as Klinefelter syndrome. So the last allosomal trisomy, it is Klinefelter syndrome. Trisomy means addition of allosome, right? Allosomal trisomy. Klinefelter syndrome. So it is trisomy means instead of 46 chromosomes, they have 47 chromosomes. 44 are autosomes plus XY. One X chromosome is added. Since Y chromosome is there, they are males. So this is common in males. Since they have X and X conditions, since they have X and Y conditions, they have feminine characters, they have masculine characters. Since they have both the characters, neither this will develop completely nor that will develop completely. That means again hypogonadism. This will not function nor that will not function. Then IQ will be low. Dyslexia means difficulty in learning. Is another symptom. Then they have gynecomastia, breast development. Because of an extra X chromosome, breast development will be there, which is called as gynecomastia. This appeared in NEET 2021 examination, and again they are sterile. Because of hypogonadism, they are sterile. So we completed allosomal disorders also. Allosomal monosomy, example Turner syndrome. In NCERT they have given the diagram. Allosomal trisomy, Klinefelter syndrome. Turner's is common in females and Klinefelter's is common in males. One chromosome is added, so instead of 46, 47 are there. 44A plus XXY is a chromosomal complement. Since XX are there, feminine characters can be seen. Since XY are there, masculine characters can be seen. But because of presence of both, hypogonadism will be there. IQ will be less. Dyslexia, difficulty in learning. Because of the presence of X and X, breast development will be there, which is called as gynecomastia. This appeared in the NEET 2021 examination and they are sterile. So allosomal disorders are sterile, they are not transmitted. Whereas Down syndrome, they are fertile but it is not transmitted. With this, we completed the chapter Principles of Inheritance and Variation. We'll meet you in the next lecture. Thank you.